Lecture today is on amino acids and proteins. The entire lecture consists of 8 objectives, and we're going to divide up into 2 or 3 lectures. So the first lecture, we're going to cover the first 3 objectives, and that is we're going to study the function of amino acids and proteins. We're going to study amino acid properties, specifically the structure, and we're going to discuss chirality and its relevance to amino acid structures. And then we're going to spend the rest of our time on this portion of the lecture with amino acid classifications. The following are some of the many functions of proteins. Proteins serve as catalysts, or these are proteins that mediate reactions. Enzymatic reactions, metabolic reactions that occur in a living system. They serve the purpose of storage and transport, such as hemoglobin. They are also structural in nature, such as actin and myosin. They do mechanical work, such as proteins that is found in flagella and cilia, microtubule movement during mitosis, and muscle contraction. These are all mechanical work related proteins. They are involved in decoding of information, such as in protein synthesis and in DNA replication and transcription. They are involved as hormones and hormone receptors. And also, the last category is that they all also serve specialized functions, such as antibodies. Amino acids are the building block of proteins. There are 20 different amino acids. And so from here on out, let me just say the following. You need to know all the 3 letters and single letter designation of all the 20 amino acids. The 3-letter designations are very straightforward. The 1-letter designation is not so straightforward. So go ahead and make you some index cards and begin memorizing this. All of the amino acids are alpha acids, and that's because of the presence of carboxyl group and amino group on the alpha carbon. At pH 7.4, the alpha carboxyl group and the alpha amino group are all ionized. And so the amino acids are referred to as Zwitter ions, 2 charges. The pKa, or the pH at which the hydrogen ion begins to dissociate. The pKa of carboxylic acid is between 1.8 to 2.5. In other words, at pH of 1.8 to 2.5, the hydrogen ion begins to come off the carboxylic acid, and the pKa of the amino group is about 8.7 to 10.7. On the NH3+, the hydrogen ion begins to come off between 8.7 and 10.7, the pHs. The amino acids have the following structures. So they all have carboxylic acid functional group, they have amino functional group, and they all have this alpha carbon and also hydrogen. We call this alpha hydrogen that is on the alpha carbon. Now, all of them have side chains that differ. So the common denominators are carboxylic, amino, and a hydrogen on the alpha carbon. But what differentiates all of the 20 amino acids is the presence of different side chains. So here's the structure of the amino acid again. The common denominators are carboxylic, hydrogen, amino on the alpha carbon. These are all common denominators, and they differ by the R group. Let's talk about chirality. A chiral molecule, you learned this in organic chemistry, is a molecule that lacks a plane of symmetry. For biological molecules, each atom in the molecule is either chiral or not chiral. Now, according to organic chemistry definition, is that when a carbon has 4 different groups attached to it, that carbon is chiral. In other words, you cannot find a plane of symmetry. Okay? The alpha carbon on all amino acids is chiral with the exception of the alpha carbon on glycine. That alpha carbon, the R group is equal to hydrogen, which means that alpha group on glycine has 2 hydrogens, and then an alpha and an amino group. Chiral comes from the word that means handedness, and so if you were to hold up your left hand and hold it up in front of a mirror, you will get the mirror image of your hand, and the two cannot be superimposed upon each other, and that's what chirality means. I'm going to skip over this. The use of the term D or L comes from glyceraldehyde, right here. Glyceraldehyde is a 3-carbon aldehyde that has a chiral carbon. Okay, right here on carbon number 2. You have 1, 2, and 3. Carbon number 2 has OH, CHO right here, CH2OH, and a hydrogen. So all 4 of these are different. So the orientation, and we're going to cover this when we get to carbohydrates, the orientation of the OH group on that chiral carbon determines whether or not that glyceraldehyde is D or L. If it's oriented to the right, it is D. If it's oriented to the left, it is L. Okay? D and L really are associated with the way that the molecule would rotate plane polarized light. If it rotates plane polarized light to the right, it is a D. If it rotates plane polarized light to the left, it is an L. Okay? And this is just the D and L designation. When it comes to amino acid, you use the same designation for amino acids. And to determine whether or not an amino acid has a configuration of D or an L, you will hold the hydrogen, you will hold the carboxylic acid on top, oriented to the top, and the amino group to the bottom, and then you hold the hydrogen to the back of the molecule. And if you can spell corn, C-O-R-N-N, if you spell corn to the right, then it's a D amino acid. If it goes to the left, then it's an L amino acid. I hope that is clear. All right? Why is that important? Well, most amino acids, with a few exceptions, that are found in nature are L amino acids. And so if you replace an amino acid in a protein from an L with a D, you're going to run into trouble. Now, why is chirality important? Well, chirality is important for the following reason, that in nature, when you generate a chiral molecule, it's going to generate both forms, the D form and the L form. Now, it turns out that perhaps one will be used and the other one will not. One will be active if they were drugs, the other one will not. Again, proteins consist of L amino acids. All the carbohydrate configuration are all D, and so insertion of a D amino acid into a protein will mess up the structure. Insertion of an L carbohydrate into, let's say, DNA will mess up the structure of the DNA or the RNA. Chirality is important when you begin to look at drugs and their activity. When we talk about drugs, we're talking about chirality or the handedness. There's a drug called thalidomide that was given for morning sickness around the turn of World War II. Now, this was used to control morning sickness in pregnant women, and it was very, very effective. But here's the problem. There was a lot of genetic deformities that occurred as a result of giving this drug to a lot of pregnant women. It turns out that the L form of the drug could be used as a tranquilizer to calm down morning sickness in pregnant women. D form, on the other hand, stunted fetal development. That's the one that caused the problem. Now, they were very careful in separating the L and the D form. But here's the problem. When you give somebody the L form, and if you make it up in a solution that deviates from the exact pH at which it was intended for, slight deviation in pH can convert the L form to the D form, and it's going to cause a lot of genetic problems. And this is a problem that they had at the turn of World War II in the 60s, after a lot of babies were born with deformities. And they come to find out that even if somebody was given the L form, alteration in pH could convert the L form to the D form very easily. So chirality in nature is very, very important. And this is thalidomide right here. This is the chiral carbon right here. The L form is the one that is used for as a tranquilizer to calm morning sickness, but it can be converted to the D form by simply slightly altering the pH of the environment. And this is what we said. Now let's go back to amino acid structures. Now, when you have these common denominators, the carboxyl group on the alpha carbon, you have carboxyl and the hydrogen and the amino group. These are all common denominators. The R is the one that differentiates between the 20 different amino acids. Based on the chemical properties of the R, you can have different classifications of amino acid, and that's what I would like to go through. There are a number of classifications of amino acid, and we're going to go through two of them. The first classification divides the amino acids up into 6 different groups. The first group is called aliphatic, and that is the R group is all hydrocarbon, straight chain hydrocarbon with the exception of proline, but they're all hydrocarbon side chain, and this is called aliphatic. Aromatic are all ring side chains. Sulfur containing, R group contains sulfur. Alcohol containing, R group contains OH. Basic, R group contains the amino group. Acidic and their derivatives, R groups contain acidic with the acidic class, and the R group contains the amide derivative of the acid. And we're going to look at all this. Let's look at the aliphatic. Look at the R groups. Aliphatic, this is glycine, R group is hydrogen, and then you have alanine, CH3, you have proline, even though it's a ring, it's in a ring structure, you have this hydrocarbon, CH2, CH2, right here. Again, valine, hydrocarbon, aliphatic, leucine, hydrocarbon, aliphatic, isoleucine, hydrocarbon, aliphatic. You can say this, since aliphatic R groups are all hydrocarbon, they are all non-polar. Okay, and we're going to get to another classification. But just think of it this way. Anytime you see the hydrocarbon, a lot of hydrocarbons, think non-polar. The second group is the aromatic, and this is very easy because you look at the R groups, and the R groups contain ring structures. The next one is all the amino acids that contain sulfur, and this consists of cysteine and methionine. They are sulfur-containing R groups. Next ones are alcohol containing, and that is serine and threonine. So they stand out. There's no way you can confuse this. The next one is basic. Any R group that contains the amino group or form of the amino group is going to be considered basic. Okay, you have lysine, arginine, and histidine. Next to the last one is acidic, and this is aspartic acid or aspartate, glutamic acid or glutamate. Now, if you were to knock off the OH and replace it with NH2, you have amide derivatives, and that is, this is aspartic acid. You knock off the OH, replace it with NH2, you have aspargine from aspartic acid. You knock off the OH, replace it with NH2, you have glutamine instead of glutamic acid. The second classification of amino acids is consists of 5 different groups. They're non-polar aliphatic, polar uncharged, aromatic, positively charged, and negatively charged. Now, this should bring back to memory your basic chemistry. And you remember the polar, non-polar, ionic, covalent. This should all bring it back to memory. And so we've covered basic chemistry. Now we're applying basic chemistry right here. So non-polar aliphatic. Anytime you see hydrocarbon, you think non-polar right here. Okay. Polar uncharged. These are polar. Anytime that you tag a electron-rich atom such as oxygen or nitrogen or sulfur right here onto the R group, you make that R group environment polar. So here we have OH, make it polar, SH, make it polar, and then of course this is oxygen, nitrogen, polar, oxygen, and nitrogen, polar. But these are uncharged. They are not ionized. Then you have the aromatic group, which is the same as the aromatic group in the first classification that we looked at. This consists of ring structure such as phenylalanine, tyrosine, and tryptophan. Next one are the positively charged. Again, anytime you see an ionic form of a molecule, that is very, very polar. So you got lysine that is positively charged, arginine that is just positively charged, and histidine, which also is positively charged. And then you have negatively charged, and that is the acid form, aspartic acid and glutamate. Remember that the R groups, the functional groups such as carboxylic acid and amino group at pH 7.4, they are charged, and we will explain why. Well, same thing with the side chain, carboxylic acid at pH 7.4, they are ionized. That concludes our lecture for the first part, and so the next part we're going to deal with the next objectives.